So we've recently published ETAC, which stands for ESMO's Tumour-Agnostic Classifier and Screening Tool. Over the last couple of years, we've seen the development of targeted agents that have a very broad activity. Some of them seem to work in all cancers, regardless of their organ of origin. We call these compounds or drugs tumor-agnostic. But it is up to now unclear from a scientific perspective what a tumor-agnostic drug is and when a drug merely has very broad activity. So the concept of tumor-agnostic is mainly regulatory, so how a drug was approved. So in the Precision Medicine Working Group, we decided to join forces and dive a little bit deeper into the topic to give out recommendations how to screen for tumor-agnostic potential and maybe help speed up drug development in the future.
As Ben mentioned, we gathered the expertise of this international group of experts to try to identify what are the minimum requirements for claiming tissue-agnostic or tumor-agnostic activity for a molecularly guided treatment option. And we thought that a good starting point would be looking into the publicly available data from the FDA prescribing information report and also the European Public Assessment reports from EMA, and assess what was the data that led to the approval of the current Group 7 indications by December 23. And at this moment, we gathered all the information from clinical trials that were submitted as a data package for regulatory assessment and tried to assess what were the common minimum requirements based on three main components: the objective response rate, the number of tumor types investigated, and the number of patients included per tumor types. And after doing this exercise and trying to identify what was the common minimum requirements, we found that obviously after all the demonstration of robust preclinical evidence and molecular biology rationale for a given drug to act into a specific molecular operation, there was a need for results from early phase clinical trials demonstrating at least 20% objective response rate in 2 out of the 3 tumors investigated and a minimum of 4 tumor types with at least 5 patients per each tumor type. And this leads to what we designed as an ETAC-S, the ESMO Tumour-Agnostic Classifier and Screener. We demonstrated in the 2 most recent tumor-agnostic indications approved in 2024 that in fact the data that was submitted met these minimum requirements that were developed based on the previous 7 tumor-agnostic indications. So we believe that this is a relatively robust test of the validity of these tools.
At first, a response rate of 20% doesn't sound all too convincing, but we have to remember that in most of these approvals we're talking patients that have exceeded standard therapeutic options, so later line patients. So a 20% response rate is good for a screening tool, but because what we aimed of achieving here is having a simple and robust tool that allows in early drug development to rate a compound whether it has tumor-agnostic potential or not. If we find out that tumor-agnostic potential is present, a company developing this asset could go ahead. If we see that there is limited or even tumor-restricted activity, this would change the course of the development of the drug. So this is the idea that we've pursued here.
Our proposal is integrating this tool after the early phase clinical trials in a drug development framework, which then work as a screening to lead drug development towards confirmatory body of evidence required to show and demonstrate real evidence for tumor-agnostic activity, not only potential. And then at the end of this flow, which is actually aligned with what we propose as a regulatory flow which could lead into conditional or accelerated approval whenever ETAC-S is demonstrating a tumor-agnostic potential. We also believe we will be able at the end of this framework to propose a definitive taxonomy classification into 3 different categories based on the interplay of the targeted effects of a molecularly guided treatment option addressing molecular aberration and the tumor-specific context and tumor-specific biology. And the interplay between these 2 dimensions led us to the definition of 3 different tiers of a taxonomy for tumor-agnostic, tumor-modulated, and tumor-restricted.
Going a little bit broader, classically in oncology, we have approved drugs in a fashion where we ran randomized clinical trials testing one drug against another. In the setting of tumor-agnostic, because we're looking at so many different malignancies, the classical development steps are somewhat skewed or complicated, and this is why we set up really this framework. And the idea is that in the end, also for regulatory purposes, we will group compounds, drugs into 3 categories. Tumor-restricted. The biology of the tumor and the organ where the tumor originated is the driver of the malignancy, and a targeted drug only works in very specific situations. We have breast cancer drugs working in that fashion. Secondly, you have tumor modulated. So you have a pathway that you see in various malignancies, and if you attack that pathway, you see responses in some, but not in all cases. We see this in pancreatic, ovarian, or breast cancer, for example, where you have common pathways. If you target them, patients will respond, but in other malignancies with the same pathway, patients will not respond. And then you have the true tumor-agnostic category, where the driver molecular alteration is really the defining factor of a malignancy, and it's not important if this malignancy originated in the lung, in the bone, or in the liver in the end. It's the molecular biology that counts and the drugs that we target to it. So summing up, we try to give guardrails and guidance in a process that defies the original and old and historic way we've developed drugs, and help the oncology community to navigate this uncharted territory in the end. To be honest, when we came up with the idea of a tumor-agnostic classifier, we underestimated how complex this issue would be, how much medical statistics and considerations would have to go into such a classifier in order to have it robust and statistically and scientifically sound. That is why we divided the project into 2 parts. We came up with these classifications that we mentioned: tumor-agnostic, tumor-modulated, and tumor-restricted, and we built the screening tool based on the data that we had at hand. In the second phase of the project, we're now aiming to define real cutoffs in between these 3 categories, and that will require some heavy scientific lifting, and Diogo will guide you through that a little.
Yeah, essentially the approach we followed used positive cases of submissions of trials that led into tumor-agnostic approved indications. But in fact, to define robust cutoffs between the 3 different categories, we also need the data from biomarker-driven basket and platform trials of with negative results and successful cases of tumor-agnostic drug development. So to gather this data, we hope to be able to develop and conduct a systematic review of basket and platform trials that assess activity of molecularly guided treatment options across different tumor types, as well as the tumor-specific trials that assess the activity of these treatment options in specific tumor types. And with this, we will have a more robust database to assess specific cutoffs between these three categories of tumor-agnostic, tumor-modulated, and tumor-restricted. We hope that this ETAC-S, the screening tool, will help streamlining the drug development process. What do you think, how do you think that this new tool could help the community to streamline and facilitate access to innovative treatments?
Yeah, thank you, Diogo, for that question. So the first thing we really wanted to do is start a discussion on what tumor-agnostic drugs actually are, because it was ill-defined. And during this process, as I mentioned, we realized that this was more of an undertaking. So our hope at the moment is that we've started a discussion, and not only within the community of practicing oncologists and scientists, but also with our colleagues in the biopharma industry, but also with regulators. So my hope would be that when we now come back with very defined and scientifically sound cutoffs, and we take all of the stakeholders in the field with us along the process, that this tool could really work in drug development with the ultimate goal to bring innovative treatment faster to the patients by streamlining this process. Is this achievable? It's going to be a challenge. Do we hope that we succeed? Absolutely.